Hi everyone and welcome to this video on how to conduct an STS page, the very first step in the Western blot. Before we go into how to conduct an STS page in the lab, let's briefly review the theory. STS page is a gel electrophoresis technique which separates proteins by size. Electrophoresis is performed with the positive anode and negative cathode at either ends of the gel. Let's first begin by talking about the STS in STS page. STS or sodium dodecyl sulfate is a very harsh negatively charged detergent. Its role is to unfold, denature, and uniformly coat the protein in a negative charge. Here we have our sample containing the protein of interest. Once STS is added, its function is to unfold and thus fully denature the protein of interest. Now that the protein is denatured or linearized, the STS proceeds to uniformly coat it in a negative charge. Because opposite charges attract, during electrophoresis, the negatively charged STS bound to our protein of interest causes protein migration towards the positive pole or anode, allowing proteins to separate by size. The larger the protein, the slower it migrates through the gel, whereas the smaller the protein, the faster it migrates through the gel. Now that we have discussed the STS part of STS page, let's move on to discussing what page means. Page simply stands for polyacrylamide gel electrophoresis. The polyacrylamide gel is made from the hydrolysis of acrylamide. Notice how the gel has a special mesh-like matrix after polymerization. This creates a suitable environment for an electric current to move protein through a gel according to its molecular weight and therefore size. Now that we know the theory behind STS page, we can now move on to the fun part, conducting it in the lab. Let's begin by taking a look at the materials and reagents. Power supply, polyacrylamide precast gel, electrophoresis tank, and running buffer. Let's now move on to the actual procedure. The precast gel should be placed with the short plate side facing inward and the flat side facing outward into the open positioned electrode assembly. Carefully place the first gel onto the assembly and hold it in place with one hand. Use the other hand to place the other plate in the assembly, also with the short plate side facing inward. Then gently squeeze the two gels and slide the assembly's green wings, one at a time, over the gels and into place. After that, place the electrode assembly with the precast gel into the electrophoresis tank. Running buffer should be added to the electrode assembly to the brim. Proceed to carefully remove the combs off the precast gels in order to expose the wells. If any wells get distorted after removing the combs, you can simply take a sharp edge and realign the wells. Remember to take your time with this step, as you want your samples to be loaded onto straight wells. It's always important to centrifuge your samples prior to loading. Now it's time to load. Carefully pipette around 15 microliters of your sample in a slow and steady manner into the well. Feel free to control the pipette with your other finger to allow for steadiness. When releasing your sample, it is important that you take your time in order to prevent pipetting error. It's now time for electrophoresis. Place the cap onto the electrophoresis tank according to its color code. Preset the voltage to around 130 volts. You can double check if electrophoresis has started by observing the formation of bubbles in your tank. That is the end of this video on how to conduct an STS page. Please like, subscribe, comment, and check out our other videos in the Western blot series.